Greetings everyone. Welcome to PG Diaries. The scientist we discuss today is Dr. John Wendell Severengaus. Dr. John Wendell Severengaus is an American anesthesiologist and a physiologist, and he is known as the father of modern blood gas analysis. He was fondly nicknamed as a master tinkerer and a human servo. Born on 6th May 1922 to Elmer and Grace Colby, Dr. Severengus has his initial training in engineering before medicine. He pursued a degree in physics from Hartford College, Philadelphia. He worked in the MIT Radiation Laboratory on radars during World War II. Later, he pursued a medical degree from Wisconsin Medical School, Columbia in 1946. He worked during an era where monitoring technology and electronics were primitive in the field of medicine. Another valuable contribution of Dr. John in the anesthesia practice is his research on the nitrous oxide uptake. They called him the human servo because he volunteered for measuring the uptake of nitrous oxide at the beginning of an anesthetic, keeping oxygen as low as 20%, and a closed system volume constant flow meters. The most notable Severingus formula was established using these experiments, and it helped to understand the rate of uptake of gases during the induction of anesthesia, and it is given as a proportion to the square root of time. The formula is still currently in use and help us understand the nitrous oxide uptake during the induction. In surgeries where the induced controlled hypothermia, as in heart and brain surgeries, they observed a need for a reliable cardiac monitor. Dr. John has invented a device called Telecor, which is a single esophageal probe monitoring device and a battery-driven and portable medical instrument. He used a 2-pound battery of low voltage complexes to prevent fire accidents in the highly explosive anesthetic environments. The Telecor used a single esophageal probe to measure the heart and breath sounds by transmitting audible signals through a speaker, as well as the patient temperature and electrocardiogram. He designed, built, and published an electrophrenic respirator and received the Borden Award for the Best Medical Student Research Project. Using his background in physics, he developed a portable respirator, which was all electronic, spark proof machine that used low voltage pulses to stimulate phrenic nerve transcutaneously. It provided a physiological alternative to the iron lung, especially during the polio epidemic. And later, the electrophrenic respirator was commercialized by the Sanborn Company. He devised the first ever remote monitoring in operating rooms. Long nylon catheters were hanged in the rooms, and gases from airways of patients were extracted. The gases are then rapidly sampled, sequentially analyzed, and provided data on a computer screen for about once in every minute. Dr. John used mass spectroscopy technique in these instruments. It was used for a strong 20 years from 1970 to 1995 up until the invention of a standalone infrared capnographic monitors. When Ted Eager developed the concept of minimum alveolar concentration, it was John Severingus who gave it the name Mach, and that naming was inspired by the Mach speed of aircraft to describe the speed of depth of anesthesia. The next biggest contribution by John Severingus in the field of medicine is his research in blood gas analysis. In 1953, Dr. John has built upon Richard Stowe's work to create a stable electrode that measured carbon dioxide via a pH sensitive glass electrode. He added sodium bicarbonate to improve the stability of this sensor. Later, he combined the modified electrode with Clark's oxygen electrode, creating a comprehensive three-function blood gas analyzer which is used to measure carbon dioxide, oxygen, and blood pH. This is the first prototype of ABG analysis machine developed by Dr. John Severengaus. He worked closely with the Radiometer Company, which has later become the manufacturer of world's best blood gas analyzers. In between 1960 to 1970s, John Severengus developed electrodes, making the transcutaneous oxygen and carbon dioxide measurements possible. They remained invaluable for the monitoring of carbon dioxide and oxygen in NICU and in sleep studies until the introduction of pulse oximeter in 1985. He developed standard equations corresponding to the oxygen dissociation curve and has also formulated a blood gas slide rule for solving the effects of pH and temperature on oxygen saturation. This has refined the mathematical models used in current pulse oximeters. Dr. Saveringas famously publicized the fact that a Japanese engineer called Takayo Aogi was the true inventor of the pulse oximetry principle and ensured that Aogi received global recognition. He also established a laboratory in the University of California, San Francisco, a hypoxic lab specifically for testing pulse oxy accuracy during induced hypoxia in volunteers, a facility that is still running and is used to provide data for the FDA 510(k) clearance. Dr. John Severengus has extensively contributed in the field of high altitude acclimatization and pathophysiology as well. In 1963, he and his team studied at the White Mountain Research Center and used themselves as subjects to measure the human CSF and its pH during altitude acclimatization. They made a groundbreaking discovery that the brain can actively regulate its own acid-base balance to control breathing when oxygen is scarce. He also showed that altitude affects blood flow to the brain and demonstrated how the body can balance the need of oxygen with changes in carbon dioxide levels. He also investigated the mechanisms behind high altitude pulmonary edema and cerebral edema, contributing to the clinical understanding of why some individuals suffer life threatening fluid accumulation at high elevations. Dr. Severingus identified a specific dog-leg bend in the ventilatory response curve where low carbon dioxide can dangerously mask the body's hypoxic drive, leading to sudden euphoria or coma, a phenomenon that caused fatal crashes for early high altitude pilots. He served as an editor for the Journal of High Altitude Medicine and Biology for over a decade, reviewing global literature on the subject well into his late 90s. Other noteworthy contributions of Dr. Severingus are his research in cerebral blood flow and cerebral acidosis that fundamentally shaped a modern understanding of how the brain regulates its environment and respiration. His group in UCSF identified chemoreceptors near the ventral medullary area. His research explored the role of carbonic anhydrase in brain decarboxylation and how it influences cerebral vasodilatation. He helped in refining the interpretation of metabolic and respiratory acidosis as well as alkalosis. To summarize the contributions of Dr. John Severengaus, he has played a pivotal role in introducing the field of electronics into monitoring of anesthesia. He is known for his big three, the carbon dioxide electrode, the first three functioning blood gas analyzer, and the transcutaneous oxygen and carbon dioxide electrode. The practical tinkerings of his inventions are the telecode, a battery-powered spark-free monitor in the era of explosive anesthetics, electrophrenic respirator, the mass spectrometry central monitoring device, and the blood gas slide rule. Even before the computers were present everywhere, John designed a physical circular slide rule that allowed doctors to instantly calculate how temperature and pH affected oxygen levels. The invisible scientific gifts he has developed for us are the standard human oxygen dissociation curve, the discovery of brain chemoreceptors, the coining of name MAC, and the pulse oximetry validation laboratory. These are my references. Thank you. Let us meet with another scientist in another session.